Xavier Muñoz from the Instituto of Microelectronics, Barcelona, and he's going to talk about monitoring light propagation through cells.
Okay, thank you very much for the invitation and everyone for assisting to this talk. I'm going to talk about our newest concept that we are now developing, Living Photonics, a way to monitor cells just using them as a waveguide, propagating the light through the cells. First of all, I would like to introduce my institute. This is the Institute of Microelectronics of Barcelona. It is located here. It's in the campus of the University Autónoma of Barcelona. It's close to Barcelona, just 20 kilometers, and this is the largest institute devoted to micro and nanoelectronics in Spain. We are around 175 employees, and as you can see, most of them are dedicated to research, but we have a big number of people working there that are technicians or dedicated to another things that are not directly related to research. And this is because our institute is quite particular. We have a clean room facility, which is huge. It's 1,500 square meters, and we have a lot of people working there. We have two lines, a CMOS line for the fabrication of products. It's more dedicated to production. And then we have a non-CMOS line for working with silicon, but also with polymers, which is more dedicated to research. This is a full equipped clean room. We have a lot of instruments. And I would like to tell you that we belong to this cluster, Micro Nano Fabs. This is the web page. And you are researchers or companies, you can apply for projects there, and they are funded. Then if you want and you have the opportunity, you can try to apply for this and get some money to do some research in our institute. In the institute, I belong to the group of chemical transducers. It's a quite big group, between 20, 25 people. And as the name indicates, it's dedicated to the development of chemical and biochemical transducers. We focus this research in four main areas: electrochemical devices. A lot of people in our group is dedicated to that. Then photonics and optical devices, also advanced materials in both cases, and the integration of all of these systems in arrays of sensors. Maybe here in this image, some of you can know Carlos Dominguez. He was the former of the group in 1992. He's more focused on the development of silicon devices, and then this is Andreu Llobera, who is working with polymeric systems. Then here, maybe there, this is me, and I'm working with materials from biological origin and also with cells. And nowadays, all of these areas we are dedicating to the development of lab-on-a-chip systems. Here, you can see a couple of examples. But before going deeper, I would like to introduce you a little about what is a lab-on-a-chip. Then, according to Nature journal, a lab-on-a-chip is a class of device that integrates and automates multiple laboratory techniques into a system that fits on a few square centimeters in size. It means integration, automation, and miniaturization, something that is fitting very well with what we want to do with micro and nanotechnologies. Maybe one of the best images regarding a lab on a chip is this one. Here we have a laboratory with many people working, doing different functions, and the lab on a chip wants to integrate these functions in different parts of the chip. In our case, the function that for us is more important is analysis, because we are experts on transducers. And we want to integrate these systems into a microfluidic device. Microfluidics are the most important part of lab on a chip and are providing them from the most important properties. Working with fluids at the microscale is providing microfluidics for laminar flow. This is a turbulent flow that we can find in the macroscale, and in a lab on a chip we have this kind of flow. What's happening when we have this flow? The only way that we have to transfer matter from one place to another is through diffusion, and this is quite slow. As you can see here in this image, even if we have different channels that go together to a single point, in some spaces there is not mixing, because they need some time to transfer matter from one place to another. This can be good or not when you want to mix, then you need to think about it. Also, in the microscale, the superficial and interfacial tension is very high, sometimes even higher than gravity. Then we can have things like this one, where we have a flow that is going against gravity. Or, for example, when we mix two samples or immiscible liquids, we have things like this. They look for the low energy structure. This is a droplet we are also using. Since in the macroscale the distances are very low, the diffusion is very high, then usually we have a higher reactivity and low response times, much lower than in the macroscale. And also, since we are using very few amounts of volumes, the consume of reagents or samples is also very small. Okay, but where it is interesting to apply these lab on a chip systems? There are many places that can be applied, but very interesting, this paper from Nature, Bibi in 2014, he looked for any paper in the world working with microfluidics or lab on a chip systems and made this word cloud. And here I realized that the word that was repeating more times is cell biology, working with cells. Why is that? The cell size is similar to the size that the channels that you can make in a microfluidic system. Then it's very logical that if you want to analyze something that you can work with the same size, it's an easy way to do it, much easier than in the macro scale. And then you can get things like this. For example, you can make patterns very small that allow you to work with a single cell. You can make this kind of grid and identify perfectly one cell and follow how this cell is evolving depending on the thing that you put on that. You can make a screening of drugs, you can make many things. Here we have a very nice example of compartmentalization. For example, here we have the place that, where we want to grow the cells. And here we have some electrodes. This is not photonics, but well. If we want to grow the cells just here, we need a very good compartmentalization, because if the cells are going here on top of the electrodes, they are not working. But here inside, we put, for example, for demonstrating this compartmentalization, we put some dyes. The red dye is just going to the reactor. This blue or purple, I don't know the color more or less, is just here in the region of sensing. Then we can do the sensing and even calibrate with a toxic analyte without compromising the stability of the cells. But all of these systems are doing more or less the same. Oh, sorry. The integration of optical components that we are doing in our group. Andrea Llobera started integrating micro-optical components inside lab on a chip systems, taking benefit from the different optical properties of air and polymeric materials, for example, in this case, PTMS. He could do this kind of lenses made of PTMS, for example, for the parallelization of the rays or for focusing the light in one point. Also, working with the same principle, for example, we could have this kind of structures, flat structures, working as mirrors, reaching total internal reflection and moving the light in the direction that we want. Even we did some curved mirrors where the angle for the tear was even higher. And just doping with different colorants to these polymeric materials, we could have absorbance filters. Maybe one of the architectures where we integrate more of these components is this one, that we call twin reactor. Here we have two reactors where we integrate many components. This is a self-alignment system. This is a channel where we introduce the optical fibers, and there we have, for example, some lenses that focus the light from here to here. Then we have a channel for the measurement of the absorbance. But we integrate two more systems, this one and this one, at 90 degrees. With here, we put an absorbance filter where we can eliminate the light that we use for the emission, and we could measure, for example, the fluorescence of the components. The main difference between these two chambers is that in the first one, we also include these blue things that are here, that are size exclusion filters. Then when we put, for example, cells, they were retained here. They proliferate, as you can see in this image, and we could measure, for example, here, the supernatant without the need of centrifugation. We measure here the pH, which is giving us information about how cells are proliferating with time. And we could do, for example, some measurements of the effect of some kinds of drugs. But all of these systems are working more or less the same. We have a cell, we put an external stimuli, a drug, change of temperature, we put glucose, anything. And after that, we measure indirectly how this cell is proliferating or is acting using the substances that they are releasing. For example, we can measure glucose, pH, lactate, or any other thing that we want to measure. Another very common way to analyze these cells is with a microscope. We can dye the cells, we can dye the nucleus, we can dye cytoplasm, everything of the cell, and measuring directly what's happening there. But when we are labeling, this is an endpoint assay, we cannot monitor. Then there is a way for doing a monitoring, but just directly, and we thought that yes. Like this. We could just confine the light inside the cells, using the cells as a dual role, because they will be the reporter, what we want to analyze, but at the same time they will be working as a real optical component, a waveguide. In this case, we will be able to directly, label-free, and contactless measure the cells, especially when we are working with visible light, because visible light is doing nothing to the cells. You work with infrared, for example, you are increasing the temperature of the cell, then they are not in a physiological conditions, and UV are killing them. But what can we measure here? We say this spectral response, but what's this spectral response in the case of a cell? For this, we need to know what's really a cell. A cell is something confined inside the lipidic membrane, and it has inside some organelles, some components. Nucleus, mitochondria. Some of them have been already analyzed in terms of optics, and they have different reflective index than, for example, the cytoplasm. Then that's for sure that we will have some scattering centers inside the cell that we will be able to analyze. But apart from that, probably we will have some proteins that will be absorbing or will be emitting, and we will have, with the wavelength, something like this, the summatory of some scattering and absorption and emission, that something that we call photonic fingerprint of this cell. And experimentally, is it possible to do? Well, in a theoretical point of view, the cells are a material that has a refractive index, an average refractive index of 1.36, which is higher than the medium. Then in principle, in this area, we could reach total internal reflection. And here, okay, we need a material support of very low refractive index. All the polymers that we are working with, they are above this value. But we were doing a lot of work about it. This is the first challenge that we had. And we found some materials that could be used. First of all, when you increase the number of fluor compounds in a polymer, you are reducing the refractive index. And in a company of Israel, they were supplying a battery of materials called MyPolymer of very low refractive index, 1.32, 1.33. And they were compatible with the technologies that we had in our institute. The problem was that even if they claimed that they were completely biocompatible, when we start working, this is not a healthy cell. They were dying. We still don't know if it is because fluorinate compounds, as Teflon, they are very hydrophobic and maybe the cells could not attach well there, or in fact they were just releasing something that was killing the cells. But this was not a good option. The second option was bacteriocellulose, paper. Bacteria can also produce a kind of paper with a lot of porosity and with a low refractive index. This paper was really biocompatible, and the problem here was technological. Here we have the paper dry. When we put it in contact with water, there is a very big swelling. It increases a lot the thickness, and we cannot control it. Then, if we want to confine the light inside the cell layer and we don't know where it is impossible to align. Finally, we found nanoporous silicon oxide. Working with Theos, which is a siloxane, and increasing the temperature, we could reach a nanoporous silicon oxide layer that it had a refractive index between 1.27 and 1.33. And this is what we decided to work with, because it was completely biocompatible. It was also compatible with the technologies that we had in our institute, and it has a quite low refractive index. The second problem, this is a more or less scale image. Cell layer is very, very thin in comparison with the optical fibers that we are using, even if the core is very, very small, because we were working with optical fibers of from 1 to 5 microns. It is difficult to align out of the laboratory. Then we need to look for another alternative, especially if we consider that the cells are not completely flat materials. They have like, when a cell is in suspension, it's like 10 micrometers, it's huge. But when they try to grow in a surface, it tends to spread around. And usually, it spreads in an irregular way. Since it has organelles inside, these organelles are quite big. Then in the region where is the organelle, there is a very big loop. Then how can we solve it? We found this paper in Nature. This is the normal configuration of a cell, but we can do something that is called polarization. It means that we can produce that the cells are just increasing their interaction with these tight junctions. Through these tight junctions, they can even transfer genetic material from one to the other. Then for sure that we have to work in polarized cells. For the alignment, we had different alternatives. The first one we call pedestal-based configuration. What we did is, with our technology, we made like a small pedestal. We put the silicon oxide nanoporous layer here and then the cells. It has like 62.5 microns, and with this we could align the optical fibers just here, as you can see in this image. Yeah, we still need to work in the laboratory, but we made small setups that we were just moving to the biological laboratories, and we could measure very well. So second alternative was this embedded fibers is the same strategy, but, well, just the opposite. What we did was to engraft the chip. In this case, it was a glass chip. And we also introduced the optical fibers. In this case, we could do the measurements, the alignment, and also use a microscope, even an inverted one, for monitoring the cells with time. The third strategy was the integration on chip of waveguides. For this, we made this design. This is the microchannel. And here we put some bended waveguides. The idea was to remove the light that was not confined. This light that was not confined, we didn't want to go to the detector. For this design, we need to make like a new fabrication protocol. We fabricate the waveguides with the thickness that we wanted. It was between 5 and 30 microns. And then we transfer these SUH waveguides to a PMMA structure, just applying pressure and temperature. In the PMMA, we already made the fluidic channels. After this transference, we just need to put a couple of layers to seal the structure, and we already have the waveguide inside, acting as a real waveguide because it was surrounded by PMMA, and then the microfluidic channel. Then, after that, we did it in the level of the wafer. And after that, we cut it, and we leave this end face very close to the surface, just for having a very easy coupling with the optical fibers. But for this, we made a chip-to-wall interface, because sometimes we cannot go to the biological laboratories and we want them to do the measurements. Then we need a very easy way for the alignment of the optical fibers. And with this chip-to-wall interface, it was very easy to put inside the chip, and it was completely aligned, as you can see in this image. This is the chip-to-wall interface. It's made of PMMA. And here we have the light that is not confined, it's going through, but then we have, I don't know if you can see very well, but there is part of the light that is confined in the waveguide and is going to the optical fiber that is going to the detector. The performance of this system was quite good, and it was very similar to what we expected from simulations. And now I will show you just some data about some measurements that we have done with cells. All of them are vascular cells from aorta. These are vascular smooth muscle cells, and these were the first one that we decided to work with, because they are not proliferating in a single monolayer, but they are much thicker. Then it was easier to confine the light inside. This was the spectral response that we had with this kind of cells over time. The first part corresponds to the medium, because in cell growing, they put indicators. This is a pH indicator, and you can see, well, this is due to the reference, but this is due to this peak of here, and this is due to this peak of here. And as you can see, these peaks are changing with time when the medium is becoming more and more and more acidic. Okay, this part is not interesting for our application in this case. But if you take a look here, you can see that some peaks are appearing with time, and these are not related to the medium. These are really related to the cells. And when we compare this spectra with this one, here we put TNF alpha. This is an inducer of some kind of response that is killing the cells. Then in this case, you can see that the response is not identical. Then, even if we were not working in optimal conditions, we will be able to differentiate between healthy and non-healthy cells, at least in the case of vascular smooth muscle cells. Then, after that, we try to work with endothelial cells, which are the important ones, because these are the ones that provide you information about the healthy state of the artery, of the vessel. They are giving the functionality to the vessel. these are working, they are proliferating in a monolayer, then this is much more complicated to confine the light, and you can see that we don't have a very rich spectrum in this case. And it was much more difficult to differentiate between the healthy and not healthy cells. But well, even though we demonstrate the living photonic concept as a new concept for analyzing the cells in a label-free contactless for monitoring, enabling monitoring, we could demonstrate the photonic fingerprint of these cells, summatory of scattering, absorption of fluorescence of different components that allow us to identify the cells and also to differentiate between different metabolic states. Even though we still have some challenges that they have to be improved with time, especially in terms of coupling of the light and also the support material. It should be improved for having a better confinement. Finally, just to, all the works need some funding, and in this case, it was funded by an European project, a LifeForce project, and also a couple of Explorer projects from the Ministry of Economy and Competitiveness. And I would like just to thank you, all of our collaborators in the biological labs, Professor Uf Simonsen from the University of Aarhus, Vicente Andrés from CENIC, and Rosalía Rodríguez from the University International of Catalonia. And thank you very much for your attention.
Thank you, Xavier. So now there's time for questions.
Thank you for the talk. It was very interesting. I would like to know, the point is that when you do this type of classification, you are making a kind of dual thing, like saying, okay, sick, non-sick. How is the degree of, how is the ability that you have to scale that? Because, there's a point in repeatability of these kind of things. Yes, you can separate both states, but where's the threshold? How you manage thresholds?
Well, we haven't done at that time because, as you could see, we have just these two positions. But in principle, we should see like a change in the spectral response from a healthy to an unhealthy. And this change in this specific peaks or this fingerprint should provide us an idea about how toxic is an agent or how ill is a patient. Because the idea is to apply it to both areas, to the screening of drugs, but also to find, for example, a way to differentiate between different types of cells or different problems.
How can you tune the response to different types of?
Yeah, in this case it will be quite easy because we can work with the concentration of this substance. Substances are not toxic or non-toxic. It depends on the concentration. Then this is what we usually do. But for this first assay, we just take a concentration that we know for sure that is really toxic for the cell. And the cells change the morphology. Because to prove the concept, we need a very big changes, especially because the confinement of light is quite tricky.
Okay, thank you.
Okay, any other question?
Hi, thanks for the talk. I have a question regarding the kind of light source you use, because I see a broadband spectrum, so I guess you're using a broadband light source. Have you explored any effects on the polarization of the light regarding this waveguide of cells?
Yeah, we haven't checked. We haven't checked, for sure. We use an halogen lamp, and we haven't checked it, sorry.
Okay, so let's thank the speaker again.
Thanks.